All right. So what you need to know for transcription, again, we're trying to take DNA and make messenger RNA. There is actually 3 steps: initiation, elongation, and termination. And so what we're going to do is go through those 3 steps. And the longest actually is going to be the first step, called initiation. This is probably the most complicated of the 3 steps. Okay, so during transcription, we're going to start it. Initiation obviously means start. To do that, we need a promoting region, promoter region. All right, so a promoter, if you don't know anything about like sports, especially in boxing, a promoter was somebody that got you excited about the sport. Hey, we need you to watch this. We want, they wanted people to bet on the fight and things like that. So this is kind of the same thing. What you have to understand, we have a DNA molecule. All of it's not going to be read. I want to make a particular protein, which is somewhere on that DNA molecule. So what happens is a promoter region is the region that is kind of trying to get RNA polymerase excited. This is where you need to attach. So what you need to know is you have this promoter region. And in that promoter region, you have what's called a TATA box. And a TATA box is a bunch of adenine, thymines and adenines together. So this big, huge group is kind of like we have stop signs today. And then sometimes stop signs that are hard to see, there will be a sign in front of that that says stop ahead. So the TATA box is kind of that, hey, you're going to want to slow down because you're going to attach down the road here. And it's about 25 nucleotides long between that and the what we call the start point or initiation site. The initiation site is where that AUG is sitting. That's where the AUG is going to actually attach. Okay, and then you have transcription factors, and these transcription factors will actually hook on to the promoter region, and they are necessary to be there for the RNA polymerase to attach. Without that, without those transcription factors, this won't happen. Okay, so that's all happening here in initiation. You got a promoter region with the TATA box and the initiation site. Okay, so that's where the start point is. That's where your start codon, AUG, is going to attach. Okay, so usually, again, TATA box is about 25 nucleotides before the actual initiation site or starting point. And then transcription factors come in, they attach, and then you'll get your RNA polymerase. And then in this case, it's usually RNA polymerase 2. There's multiple RNA polymerases. All right. And so that's what this is saying. So elongation, RNA polymerase 2 is the actual polymerase that is adding nucleotides to the 3' end. Now, the good news or bad news, there's not a leading and a lagging strand on this. We're only, messenger RNA that's being built is only single-stranded. So we're only copying one side. We're only copying what's called the template side and just kind of ignoring the other side. But again, it's going to be built very similar to the leading strand. Now, we don't need a primer because it's RNA, not DNA. And because it's not a lagging strand, there's no fragments, nothing like that. And it's going to be built continuously. And we will just continue to add pieces of mRNA until we get to the 3rd step, which is termination. And that's when one of the stop codons are brought in, UAA, UAG, or UGA. Once that happens, you now have your messenger RNA. It will separate out. And what I do want you to know is that unlike before, where you have DNA unzipping itself and then being copied for new DNA molecules, once this thing is copied, it will actually just go ahead and shut itself back down. So the DNA comes back together. So again, those are the steps: initiation, elongation, termination. All right, so the messenger RNA that just has been built in the nucleus is not what leaves the nucleus. So there's what we call RNA processing. There's really kind of 4 steps, and I've got 2 on each slide. What has got to happen is on the 5' end, there is a modified guanine nucleotide cap, 5' G3P. G3P cap. I don't like to use G3P because we use that for sugar, but a 3-phosphate guanine cap. And then on the other end, there is a 3' poly A tail, a bunch of A's attached to the back end. Okay, so those 2 things that are happening are really thought to be to get through the nuclear pores. This RNA has to go through the pores, and it believes that these 2 caps actually helps it get through the pores. All right, the other thing is RNA splicing. The RNA the messenger RNA that's built is not the messenger RNA that actually leaves the nucleus. There are things called introns and exons, and I kind of want you to get this concept. When I first started learning about this stuff, there was no introns and exons. I mean, they always existed. We just had different names. And it was basically introns were called junk DNA. We didn't know what they did. They weren't transcribed into proteins. We assumed they were garbage. That is not the case. We found out now that they do things, and we'll talk about that a little bit in a minute or 2. But here's what I want you to know. Introns are put in the trash. Think of them. You can kind of think of them as junk. They're not going to be taken out of the nucleus. Exons, on the other hand, are the parts that are going to be read for the proteins, and so those are actually spliced together. So I want you to think introns are cut out, they're spliced out, and then the exons are actually, I should say spliced out, are excised out, and then the exons are spliced together. And it is the exons with the guanine cap and poly A tail that actually leave the nucleus. So what I need you to know, it's a spliceosome, again, a protein, small nuclear ribonuclear proteins that actually do this process. So it's a spliceosome that comes in, grabs onto the messenger RNA, bends it into the right format where the exons can be spliced together and the introns can be cut out and removed. So what the hell are introns? What we're finding is they're used for gene expression that we'll talk about later. Depending on what intron is in front of an exon will determine what happens. A single gene can code for different polypeptides depending on the intron it is associated with. And then there's this idea of domain structural and functional regions may allow exon shuffling. And so what we're finding out now is the introns may actually lead in how these things are read over time. We think that they might have been involved in, or maybe involved in allowing us to adapt to what's going on. There is talk that introns were actually at one time functional, and they are no longer functional. They're remnants, although again, there's still regulatory things like that. So lots of different reasons to have introns still. Bacteria have something called a plasmid, another section with extra DNA in it that they can move. This is, ours is incorporated into our chromosomes, kind of, I guess. It kind of does the same thing. So lots and lots of study now being done on introns. And so we will stop that.